Okay, so I told you the difference between the two terms, sanitization versus sterilization. Sanitization is way to remove the bioburden, but sterilization is complete elimination of bioburden, right? Then for the sanitization, what do you use to remove, reduce the amount of bioburden? So you use chemicals that are considered antiseptic, or disinfectants, or sanitizers, right? So examples are alcohol, like ethanol or isopropyl alcohol. Those are good to reduce the number of bioburden, because many of bacterial cells will be killed if you expose bacterial cells to this alcohol-based solution, about 70% concentration. And then other possible sanitizers are phenolic compounds, quaternary ammonium compounds. There are brand names that are well known, like Lipase or Birex is all good brand names. Yeah, they are also effective in killing bacterial cells. Another kinds are bleach, right? Those are very effective sanitizers. So chemical name for bleach is sodium hypochlorite. Okay, so it's very strong, as you know. Clorox is a good example. It can kill bacterial cells very efficiently. Not only it can kill the bacterial cells, they are resilient life form. Those are spores. Spores are also killed by bleach, 5% solution. And then another effective one is called hydroperoxide, 3% hydrogen peroxide, or BHP. It's a vapor form hydrogen peroxide. What VHP is, vapor form. They can also kill bacterial cells very effectively, and spores as well. So you remember that filling machine I told you in the picture before? So when this filling machine should be sterilized at the end, okay, and you use vapophage hydrogen peroxide to kill whatever that is still in there in the space. Okay, it's very effective. So these two are the best, actually, the bleach and hydrogen peroxide, because they are sporicidal. They can kill spores as well as bacterial cells, but these are effective in dealing with the bacterial cells, but they don't kill spores. Okay, so you may have to combine two of them together, maybe one sporicidal and one of these things like ethanol or something like that. Okay, so you can sanitize the environment or your hands and your workbench, work area. Okay, but still somebody has to come in and validate and see if your clean room is really clean. We call that process as environment monitoring. Okay, your environment, clean room area should be monitored. Okay, so it's evaluating process that should be taking place daily. Somebody, maybe QC technician, will come in and take some environment monitoring activities, some samples. So by taking samples and measure the amount of CFUs in there, they can measure the cleanness of your classified area. But they cannot test all the clean room area, right? So they'll have to take some representative samples, of course. So those samples will show you the snapshot of different area. So your sampling method should be good. So that the environmental monitoring method should be compliant to the FDA guideline, of course. It should be meaningful, manageable, and defendable. So meaningful, it should represent the snapshot of the entire process, entire space. Manageable, you should have manageable number of samples taken daily. And then defendable, what if there's an audit from the FDA? Your environmental monitoring method should be defendable from their questions. Okay. What kind of things are typically being measured through your environmental monitoring? Of course, air samples will be taken, and you will measure non-viable particles that are floating in your air sample. Okay, and then another air sample will be taken, and then you will measure the viable particles. Okay, and so you will see not only the air sample, you can also take samples from cleaned up area like floor, wall space, your workbench. Okay, because those areas should be all sanitized. You can measure the efficacy of your sanitization process. Personnel as well. When people are gowned up aseptically and walk in there, did they really not contaminate their gowns during the gowning process? They'll take some samples and measure if they are really clean when they gowned up. Water, of course, is important. Water is used inside of a clean room area. You need to make sure that water meets the UFI requirements, UFI quality. We talked about the water quality, right, in the previous class. So if you are measuring the air quality, of course, like I said, non-living and living particles, they will be all measured. Okay, so then what do you mean by living? Those are like microorganisms that can come and reproduce. We call those viable particles. Okay, they should be able to come back to life and they can reproduce. Okay, so you need to have a special device that can capture viable air particles, and you have to measure certain ways. You measure the results should be quantitative, meaning you have to have CFUs per whatever the size of the sample. Qualitative means what? You should be able to identify what is that maybe a particular species of bacterial cell, so you can tell those infectious cells that can contaminate your product. If they are certain species of bacterial cell, where would they be coming from? Okay, so those are the qualitative data you need to have as well. Non-viable air sampling. So if you have particles in your air, there may be some non-viable particles in there, just as what, like fibers or human hair or whatever, those kind of things. You also need to measure those. Okay, like 0.5 or 5 micron particle sizes should be measured. So there are devices that can measure those. Okay, so we call these as viable air samplers. So they take air samples, and then this device has this petri dish full of growth medium. So the air samples will filter through this growth medium, and then you can get these plates out and incubate it, and you can see how many CFUs you can find. Okay, and these are non-viable air samplers, air monitoring, air particle counter. So these machines can take in air samples, and you can measure by shining the laser beam. You can count the number of particles in different sizes from your air sample. Okay, so viable particle detection, like I said, there are different ways of doing it. A passive way will be you can set up a setter plate, a plate like this, is set on a corner where there is less human traffic, and with the lid open, and then whatever the particles floating there can be settled on the plate. That's why it's called setter plate. Okay, and you expose about 1 hour time frame, and you come back and put the lid back and incubate it and see how many particles will come back to life. So count CFUs from your plate. Active way, active sampling is use a machine like this. These are active sampling method because they have pump. They will draw in the certain amount of air. Okay, and that air will be touching the nutrient media like these plates. Okay, so automatically those living particles will be inoculated on the petri dish. So you're gonna grow those cells on the petri dish for about a couple of days, and then you're going to count them. Okay, so that's a viable air sampling method. So FDA guideline for aseptic processing. So federal standard, ISO standard, so if you are working in a clean room, class 100 in the federal standard or ISO 5, the particle number, okay, larger than 0.5 micron size, you should have less than that many in cubic meter space, 3520. Okay, if it is in maybe 10,000 federal standard and maybe ISO 7, it's about 100 times more. Okay, is allowed. Okay, and then if it's a class 100,000 or ISO 8, maybe 10 times more you can have. Okay, so those are the particles, regardless of living or non-living. But if it is a living viable particle, they said you can only have one viable particle in the cleanest environment. Okay, ISO 5 and federal class 100, just one living viable count is allowed. In the 10,000, only 10 CFUs are allowed. class 100,000, only 100 viable counts are allowed. Okay, so these are good guideline. So if you have more than 1 in your cleanest environment, there's a problem. If you have more than 10 in your federal standard class 10,000, you are in trouble. Okay, so this is a design of that particle counter that just measures any kind of particle. So it takes in the air, sample of air, and then the air is going through this chamber where a laser will shoot through that air. Then this machine can count the number of particles that are reflecting the laser. So, and the air comes out through this way. So those are called particle counters. Something like these are all particle counters. Okay, so they can be detecting the particles through remote sensing mechanism, or it could be a handheld device. Or I showed you some picture of these. These are also particle counters of different kind. Okay. Okay, so we talked about quantitative measure. So these machines will measure number of particles per your air sample, or number of CFUs in your air sample. Then when you try to qualify what kind of contaminants are found from your samples, and how do you identify what kind of cells do you have in them? Okay, so this is the method that you can use. So if you're a microbiologist, there are ways of identifying microbial species using different types of media, selective media, and that has different nutrients. So it depends on the genetics of microbial cells. They are able to grow on certain medium, or they are not able to grow on certain medium because each medium has different nutrients or different amino acid or different sugar. Okay, so based on those selectivities of the cells, you can score or you can classify bacterial species down to the species level. Okay, it's a bacterial classification system, conventional method. So think about Gram staining method. So Gram staining can group bacterial cells into just two groups, either Gram positive or Gram negative. So Gram positive will be showing you kind of pink color with the Gram staining method, and Gram negative and Gram positive will give you a purple color. Okay, so the color-based assay, because their cell wall structure is different. Okay, so that's one way of classifying them into two groups. And then there are different media, differential media that has different nutrients, can also change their color, depends on cells are utilizing the media or not. And by scoring all these, you can tell what species of bacterial cell you have. Okay, so the conventional method, you will use a bunch of different medium and you can get the score. And simpler way of doing that is using something like this. It's called API strip. And so in a strip, you have differential media of different kinds. So you're going to add your cells that you identified from your sampling method into all these different media and see how those media color will change. Okay, so there's a score sheet. Okay, if you compare the result, did it change? Did it develop color or not? No color development. At the end, it'll give you the score number. And if you go to their website, the website will tell you, based on the score, what species of bacterial cells you are dealing with. Okay, so it's kind of a pretty cheap method. It costs only $6 per strip. You can use that. Another way of doing that is more expensive method. If you have a machine like this, which may cost $100,000, this machine will do that scoring method for you. These are called automatic bacterial identification system made by Vitek Biologs. Okay, so your bacterial samples will be analyzed through this machine, and the machine will tell you what strain you have. Okay.